Hello, Grade 8. This is the Biology Exam Review Worksheet for Semester 2, the midterm exams at the International School of Innovation.
Let's start with part 1. It's about immunology, the immune response, activity 1 about the self and non-self. So where do we have peace? Beside the people that we know, that we feel comfortable with. What should have they done? What is the role of an army in a kingdom? What do you think the army in a kingdom is going to do? It's going to defend this kingdom, defend the people of this kingdom, and protect them. What about us, humans? What do we have to protect us? The real answer is, are we living in a safe environment? If it is for me, environment is contaminated with viruses, bacteria, pathogens, and a lot of other harmful substances. So, to start with, we have something known by the immune system. What is the function of the immune system? The immune system is the army that protects our body against the harm around. It's the army that protects us against bacteria, against viruses, against parasites, and others. So, the immune system, what is the role of the immune system? The role of the immune system is to defend the body against any possible invasion by bacteria, viruses, parasites. Moving to, what is immunity? To start with the definition of immunity, it's the body's ability to respond to foreign attacks by parasites, bacteria, and viruses. This ability is called by immunity. Moving to, what is the immunology? Immunology is the science that studies immunity. It studies the immune system, the cell of the immune system, the organs of the immune system, and the mechanism of action, how it protects the body. Is the army made up of one single cell or one single unit? If you see here, we have airplanes, for example, helicopters, 16 for example. We have terrestrial, and we have the one that are used to protect us from the sea, from the land, and others. So the cells of the immune system, they are mainly composed of different types of cells. The parts of the immune system, we have lymphocytes, we have monocytes, we have neutrophils, and others. So these are the cells, the tissues, the organs that are responsible for our immunity. Here we have the Lebanese army and the Israeli army. The Lebanese army is going to attack any Israeli army that's going to enter our country. This is called a non-self. While in this figure, the Lebanese army is going to protect these Lebanese citizens, so it will not attack the Lebanese citizens. Here we have the army of France, for example, is not going to attack the president of France, which is Sarkozy, so it will not attack the president of France, while the Lebanese army is going to attack the French army whenever they enter the country. So what is the big question? What determines whom the army should attack? What is the markers, or what are the biological identities that allow us to protect against this and not to attack others? Would Ali's immune system attack Ali's lungs? Why and why not? This is the function of the immune system. So Ali is having lungs. So the lungs, they are classified as self. Ahmed needs a lung here. Some problems in the lungs, it needs a lungs. So would Ali donate his lung to Ahmed? What would Ahmed's immune system do? This is the big question. So immune system defends, attacks citizens or foreigners. So the big question is, what determines this? So the immune system, defends its own citizens, and it attacks any foreigner. It defends the citizens and attacks the foreigners, any foreign body. This is the citizen, they are classified as self, while the foreigners, they are classified as non-self. So self, non-self. What determines what's self and what is non-self? What is self? Self is all that belongs to one's body, like my lungs, my stomach, my intestine, my eyes, and others, while the non-self is something that does not belong to one's body, like my friend's hand, my friend's nose, my friend's mouth, my friend's skin. All these are non-self. They don't belong to any part of my body. So here, which would the immune system attack, the self or the non-self? Sure, they will attack the non-self. Here we have self or non-self. Sure, they will attack the non-self. Moving to, let's develop Freyr's model for the self and non-self. How does the immune system distinguish self from non-self? As such, they distinguish us from HLA. What is HLA? HLA, the definition is the human leukocyte antigen. This is the name human leukocyte antigen, HLA, found on the surface of nucleated cells, the cell that has nucleus. So it's a biological identity marker that helps the immune system to distinguish between self and non-self. So as such, HLA, the cells of the same body and those of the identical twin, they have the same HLA. My body, the cells in my body, they have the same HLA. The cells of my identical twin is going to have the same HLA, while the cells of different persons, they are going to have different HLA. Each person has a specific HLA on the surface of nucleated cells. Note that red blood cells, they don't have a nucleus. They are called anucleated cells, so they don't have HLA on their surface. This you have to note this. Note also, identical twins, they have identical HLA on their nucleated cells. So let's develop Freyr's model for self and non-self using HLA terms. All that belongs to one's body and have the same HLA is called self. Example, it's my hand. This is an example. Non-example, it's my friend's hand. It's classified as different from the self. It's classified as non-self. So non-self, all that does not belong to one's body and has different HLA. Non-self, example, my friend's hand. Self is the opposite of non-self. It's my hand. So what is a graft? Graft is the transplant of a tissue or an organ from one side to another in the same body or from one body to another. So the transplant of a tissue or an organ from one side to another in the same body or from one body to another. Types of grafts. We have 4 types. Let's start using Freyr's model to define autograft. The autograft is the graft that's transferred from one side to another within the same body. It's called autograft. Autograft example, from my hand to my thigh within the same body. Why? The opposite of autograft, from my hand to my friend's hand. This is not an autograft, this is non-example. Okay? This is the first type, autograft. Isograft is the graft that's transferred from one person to his or her identical twin. So news can be transferred to his identical twin. So whenever we have isograft, we should have identical twin, not only twins, identical. An example is a news can be transferred to his sister twin. Allograft. What is allograft? It's the graft that's transferred from one person to another in the same species. the same species. For example, from Maya to Rolla, they are the same species, human being, from a cat to another cat. Okay, while from a cat to a dog, from a cat species, this is another species, this is the dog, this is another example. This is the xenograft. Xenograft, we graft from one animal to another from different species. They are not belonging to the same species. Example, from a cat to a dog, different species. While from Maya to Rolla, this is considered as allograft, meaning the same species. Notes: what affects the fate of the graft? The fate of the graft, whether it's success or failure, depends on the similarity of HLA molecules between the donor and the recipient. What is the donor? It's the one that donates, gives the body organ or other, while the recipient is the one that receives, that takes this transplantation.
Okay, let's move here to activity 2 about the immune cells. We have blood. What are what is blood and what does it contain? Blood flows everywhere through the human body. Sure, we cannot live without it. The heart pumps blood to all our body cells, supplying them with oxygen and food. Also, blood carries proteins and cell fragments. They are called platelets that let blood clot where there has been an injury. Blood cells, they are divided into two groups. They are divided first into something known by red blood cells. They have another name, erythrocytes, and white blood cells, they have another name, leukocytes. So red blood cells, they are small in size, they are many in number, while they are white blood cells, they are large in size, few in number. They are red in color, while the white, they are not red in color. The role of red blood cells is to transport oxygen and carbon dioxide, while the role of white blood cells, they defend the body. They act as immune cells. They are red blood cells, they are anucleated cells, meaning that they don't have a nucleus, while the white blood cells, they are nucleated, meaning that they have a nucleus. Now we're gonna move to this figure. This figure shows the red blood cells. They are 5 to 6 million cells per milliliter. They are white blood cells here, and they exist about 5,000 cells per milliliter. These are the red blood cells, the blood smear, and these are the white blood cells. This is the red blood cells. This is the figure showing red blood cells. Moving here to this figure, it shows different cells of the immune system. We're going to know them one by one. Cells involved in immunity. First one, it's called the monocyte. They are characterized by their horseshoe-shaped nucleus. So the cells involved in immunity, first we have monocytes. Monocytes, here the figure of monocyte. This is the horseshoe-shaped nucleus. It looks like horseshoe-shaped. This is the cytoplasm, and this is the cytoplasmic membrane. What are the properties? They are large in size. They have large horseshoe-shaped nucleus. They flow in blood vessels, and they can cross the capillary walls. They can mature into a macrophage. What's the role of them? They are having a role in phagocytosis, meaning that eating other cells. Other types of cells, they are called granulocytes. Granulocytes, the same as the monocyte, they have the cytoplasmic membrane. They have, instead of horseshoe shape, they have a multi-lobed nucleus, and they have granules in the cytoplasm. What are different properties? Also, they are large in size. They have a multi-lobed nucleus. They have granules in the cytoplasm. They flow in the blood vessels. They can cross the capillary wall, and they have a role in phagocytosis, meaning that they can eat other cells. Here we have lymphocytes. We have two types of lymphocytes. We have B lymphocytes and we have T lymphocytes. Let's start with the B lymphocytes. They are large in size. They have large rounded nucleus. This is the nucleus, large rounded nucleus. they have small cytoplasm and they have a role in secretion of antibodies to kill the non-self. B lymphocytes, this is the figure, cytoplasmic membrane. Here is the large round nucleus. Here we have small cytoplasm and they secrete antibodies. With the other type of lymphocytes, this is the T lymphocyte. T lymphocytes, the same as B, they are large in size, they have large round nucleus, they have small cytoplasm. While the role of T lymphocytes is different from the role of B lymphocytes. They are killer cells, meaning that they use their T cell receptors, these are the T cells receptors, TCR, to kill the non-self. They don't secrete antibodies. This is cytoplasmic membrane, large round nucleus, small cytoplasm, and this is the TCR, T cell receptor. Molecules involved in immunity, we have the antibodies. They are Y-shaped proteins. This is the figure of antibodies, Y-shaped proteins. They are secreted by the B lymphocytes into blood. Antibodies, they are specific. What the meaning of this? That every antibody can bind a specific non-self antigen. So this is what I'm talking about. Every antibody can bind to specific antigen. This is antibody for antibody. Antibody 1, for example, it has a round shape, it can bind this. This is triangular shape, it can bind this. This is how rectangular, it can bind this. So every antibody binds to specific antigen. Let's start now by this application. In order to identify the organs responsible for the production and maturation of the different immune cells, 5 different groups of mice were subjected to different treatments, summarized in the following table. So what is the aim of this experiment? What is the aim of this application? Is to identify the organs responsible for the production and maturation of different immune cells. Group A of mice has no treatment. We see the presence of mature B and T lymphocytes in the blood vessels. So group A, no treatment. We see the presence of mature B and T lymphocytes. Group B, we have irradiation with X-rays. X-rays is known to destroy the bone marrow. We have absence of mature B and T lymphocytes. So no mature B and T lymphocytes in the blood vessels. Group C, we did irradiation with X-rays and followed by grafting of a bone marrow. As I said, X-ray, they destroy the bone marrow, and then we graft a bone marrow. We see the presence of mature B and T lymphocytes in the blood vessels. Group D, we have thymus ablation, meaning that we remove the thymus, and then we did irradiation with X-rays, followed by grafting of a bone marrow. We see no thymus, so presence of mature B lymphocytes and immature T lymphocytes. The conclusion here is that there is no thymus, so no mature T lymphocytes. The thymus is responsible for the maturation of T lymphocytes. Group A, we remove the thymus, so no maturation of T lymphocytes. Irradiation with X-rays, there is no bone marrow, we destroy the bone marrow, followed by grafting of thymus. Okay, what's the result? We are coming to know. First, note first one that radiation, as I said, destroys the bone marrow. Question, what is the objective or the purpose of these experiments? We explain this. What is the role of group A of mice? It's a control group used to explain and compare different experimental results. We have to interpret. What the meaning of interpret? It's analysis plus an indication. And finally, we have to predict the results of experiment E. And we have to explain. First, what is the role, or what's the aim, is to identify the organs responsible for the production and maturation of different immune cells. For example, in the exam, if I told you what's the aim, you have to pick out the aim from the text or from the figure. So I said you have to identify, for example, specify, classify, all these terms at the line of Wi-Fi, you have to justify. For 2, what is group A? Group A of mice doesn't receive any treatment, so you have to say they are control group used to compare and explain the different experimental results. Fourthly, you have to, without any treatment, mature B and T lymphocytes are present in the blood vessels of group A of mice. What does this indicate? This is analysis, and this is the indication. This is the meaning of interpret. This indicates that mice can produce mature B and T lymphocytes in the normal state. Second, after irradiation of mice of group B with X-rays, so destruction of the bone marrow, no B and T lymphocytes are found. Meaning that the bone marrow is responsible for the production of B and T lymphocytes. Since the bone marrow is destroyed by X-rays, so there is no B and T lymphocytes. Moving to experiment 3, after irradiation of mice of group C with X-rays, we did irradiation with X-rays, so we destroyed the bone marrow, followed by grafting of bone marrow to give it down in bone marrow. Natural B and T lymphocytes are found. What does this indicate? This further indicates that the bone marrow is responsible for the production of B and T lymphocytes. Group D. Group D say that after thymus ablation, followed by irradiation with X-rays and grafting of bone marrow to remove the thymus. Thymus ablation, removal of the thymus. Mature B lymphocytes and immature T lymphocytes are found in the blood vessels. What does this indicate? This indicates that the bone marrow is the site of maturation of B lymphocytes. Yet, the T lymphocytes mature inside the thymus. Present in Group E. Group E, we have total absence of B and T lymphocytes since we destroyed the bone marrow. Okay, this is due to the fact that upon irradiation with X-rays, the bone marrow will be totally destroyed. As such, no B and T lymphocytes will be produced at all, even though we grafted in thymus again. Okay? Even in the presence of thymus, as I said, even in the presence of thymus, no T lymphocytes are there to mature in. We don't have the factor for the production of T lymphocytes. How do they mature? We don't have T lymphocytes. There is no maturation of T. So here we have a question: what are the organs of production and what are the organs of maturation? Bone marrow, thymus, and we have primary immune organs, we have secondary immune organs. What are the primary? The organs of production and maturation of immune cells. Secondary, they are the sites of the immune response. As said, immune cell is primary, secondary, bone marrow, and thymus. While secondary, there are the lymph nodes and spleen. Bone marrow, production of all of immune cells, B and T lymphocytes, and responsible for the maturation of B lymphocytes. So bone marrow, maturation of B. While thymus is the maturation of T lymphocytes. So the production of all immune cells is for the bone marrow, produce B and T lymphocytes, while the maturation is only for B. While thymus is responsible for the maturation of T lymphocytes. Lymph nodes, they are the action, or they are the site where immune response against tissue infection, while the spleen is where immune response against blood infection. This figure shows some antibodies, IgG, and so on. So we are going to move to the natural barriers of the human body. Natural barriers, we have first skin. So the natural barriers, first they are the skin. We have, other than skin, we have the eyes, mainly they are the tears. We have other forms, for example, sweating. We have the tears, they are natural barriers. It's the tears. We have the ears, they are considered as the natural barrier between natural barriers, meaning the waxing and so on. We have the waxing, as I said, so we don't have to use these sticks. We have the mouth, has a ring, and we have stomach. We have the sweat and the sweating glands, as I said before. The natural barriers we form The first line of defense against any bacteria, any parasite, any infection, any virus. So what would happen if these natural barriers were broken? If they pass through the first line of defense, what will happen? If the first line of defense is broken, we're going to have first infection. What is infection? It's the invasion and the multiplication of pathogens in the body. Second, what's the meaning of pathogen? It's any non-self organism that infects the body, bacteria, viruses, and so on, and is capable of causing a disease. Inflammatory response or inflammation is the immune response against infection. This is the second line of defense. Whenever we have inflammation, so the first line of defense is broken, and here we have the second line of defense. What are the four signs of inflammation? First, we have redness, heat, edema or it's called swelling, and finally we have pain. 1. Redness. What is the redness? It's due to the dilation of the blood vessels and the flow of more blood to the site of infection. This will cause redness. 2. We have heat. Heat is due to the dilation of blood vessels and the fast movement of blood cells, the fast flow of blood cells to the site of infection where the immune response starts. 3. We have, this is what I know about the heat, this fever had to combat bacteria due to the fact that pathogens, they can survive high temperature. 3. We have swelling, or it's called edema. It's due to the flow of immune cells across the capillary walls toward the infected site. 4. We're gonna have the pain. Pain, as I said, is due to the swelling that creates more pressure on the nerve cells in the surrounding of the infection site, so this will cause pain. Note, the first cell to cross the capillary walls are the monocytes and the granulocytes. Why? Because they have phagocytic ability. They are called phagocytes. Note, when the monocyte crosses the capillary wall, it becomes a macrophage. This is the monocyte in blood vessels, and this has become a macrophage in the affected site. Characteristics of inflammation: maybe it's instantaneous or maybe non-specific. Instantaneous start immediately, while non-specific doesn't depend on the identity of the pathogen. Instantaneous is start immediately, and non-specific doesn't depend on the identity of that pathogen. Now, this is the figure that showing the macrophage. The macrophage during the process of phagocytosis, while injecting the bacteria step by step, the macrophage extends forward to capture the bacteria. Second step, the bacteria are captured and trapped within the extension of the macrophage. And third, the bacteria which are trapped in the macrophage membrane are observed one by one. This phagocytosis, the macrophage which is yellow in color, this one is digesting the bacteria that are blue in color. The definition of phagocytosis. What is phagocytosis? Phagin means that eat. Cytose is the cell. Phagocytosis is eating other cells. It's the non-specific immune response ensured mainly by granulocytes and macrophages. They have phagocytic ability. These cells, they come near the microbe, adhere to them, engulf, and digest. In the exam, I'm going to give you three figures. Sure, they are going to be more clear than these ones. So 3 figures. I'm going to tell you, for example, what does this figure represent? Mean this process or mean this phenomena? For example, I'm going to tell you order the different steps. I'm going to give you these steps in different order. For example, I'm going to put this number 3, this is number 1, this is number 2. Order them. Question number 3, I'm going to say explain each step. Each step you have to explain it. That is the first step, the macrophage come near the bacteria. Second, the macrophage adhere to the bacteria and surround them. Third step, the macrophage engulf the bacteria and digest. So the first step, the phagocytes form the pathogen. Second, the phagocytes adhere to the pathogen and gets in direct contact with it. And the third step, the phagocyte engulf, ingest, and digest the pathogen. This figure showing a macrophage white blood cells resulting in leukemia. Phagocytosis, which is a real one under the microscope. Phagocytosis is a non-specific immediate response against infection. Main actors, we are the phagocytes, we are the monocytes and granulocytes. The monocyte, we have the whole nucleic nucleus, and the granulocyte, we have a multi-node nucleus. Phagocytosis, the site is non-specific immediate immune response against infection. The main actors are the phagocytes, monocytes, and granulocytes. What are the nature drivers? They are the cures, acidification, skin, serous skin, mucus, ciliated cells, and gastric juice. Now let's start with the worksheet. Let's start with exercise number 1. Indicate the true statement or statements and correct the false one or ones. If it's true, you have to say true. If it's false, you have to say false, and you have to write it in a complete answer. All human body cells, they have HLA. Sure, this is false. All nucleated human body, all nucleated human body cells have HLA. Since we have unnucleated, they don't have nucleus. Example about this, the red blood cells. Isograft is the graft carried out between twins. False. It's a graft that's done between, not twins, between identical twins. In the exam, you have to write a complete answer, okay? 3 brothers share high similarity between their HLA molecules. These they are brothers, so this is true. Xenograft is done between different species. This is also true. Allograft and autograft are accepted by the immune system. False. You have to say auto and isograft. Autograft and isograft are accepted by the immune system. Thymus is the site of maturation of B lymphocytes. Let's say thymus is the site of maturation of T lymphocytes and not B. Monocyte, they have a horseshoe-shaped nucleus. This is true. Now we're gonna move to exercise number 2. Complete the table below. Autograft is the graft done within the same body or within the same individual. Result, this is tolerance or acceptance. Tolerance. Isograft between 2 identical twins. Individuals of different species, we have here xenograft, and mainly it's rejected, so we have rejection. Allograft, different individuals of the same species, and mainly it's rejected. Question number 3. A person with blood group A needs an immediate blood transfusion. 3 donors. Here we have 3 donors are available, donor 1, donor 2, and donor 3, but we don't know their blood groups. A blood test was done for 3 donors and the results are shown below. This dark one is saying no agglutination, so no binding of antigen and antibody. Agglutination, this dotted one means agglutination. First one, identify the blood group of each donor. Which one of the above donors can give for the patient of blood group A? Justify your answer. Identify is the rhyme of Wi-Fi, so you have to justify. So after adding anti-A and anti-B antibodies to sample taken from donor 1, it shows agglutination in presence of anti-A and anti-B. What does this show? It shows that it contains antigen A and antigen B, so the blood group is AB. While the second one, it shows agglutination in the presence of anti-A and not in the presence of anti-B. That means it contains only antigen A, so the blood group is A. Here, it shows no agglutination, neither with anti-A or anti-B, so it doesn't contain anti-A nor anti-B, so the blood group is O. Which one of the above donors can give for the patient? This one. After adding anti-E and anti-B antibodies to blood sample taken from the donor 1, it shows agglutination in the presence of anti-A and anti-B, showing that it contains both antigens A and B. The blood group is AB. After adding anti-A and anti-B antibodies to blood sample taken from donor 2, it shows agglutination in the presence of anti-A and not in the presence of anti-B, meaning that it contains only antigen A, but the blood group is A. Blood group donor 1 is AB. So after adding anti-A and anti-B antibodies to blood samples taken from donor 3, it shows no agglutination in the presence of both antibodies A and B, which means that it lacks both antigens, and the blood group is O. Second part, which one of the above donors can give for the patient with blood group A? Donors 2 and 3. Donor 2 is blood group A. Donor 3 is blood group O. No is known as a universal donor. So donors 2 and 3 can donate to a patient of blood group A who carries anti-B antibodies, since donor 2 have the same blood group A and carry the same antigen A, and donor 3 has no antigens A and B, meaning that no agglutination can occur. Okay? Moving to question number 4. Question number 4, in an attempt to study the factors that lead to graft rejection, the experiments schematized in the adhesion figure are performed. Rabbits 1 and 3, they are identical twins, 1 and 3, while rabbit 2 is not related. Each of 3 RNAs means a skin graft. Name the type of graft made in each of the experiment A, B, and C. A between the same individual, so it's called autograft. So A between the same individual, it's called autograft. B between 2 individuals of same individuals, they are different with each other, so they are allograft. Same species, different individuals. And we have C between 2 identical twins is called isograft. Moving to part 2, using your acquired knowledge, indicate the fate of each. Indication, you have to say whether it's accepted or rejected. I don't need justification here because I said indicate. So indicate the graft in experiments A and C is accepted, while in B it will be rejected. Moving to part 3, the graft from rabbit 2 to 1 to 1 is rejected, except A is rejected. 2 to 1. 2 to 1, we have low compatibility in HA molecules between rabbits 1 and 2, which are not related. The cells of the graft from 2 will be rejected as non-self, will be recognized as non-self, and will be rejected by the immune cells and attacked. Moving to part 4, we ablate the thymus site of maturation of T cells from rabbit 1. And then we repeat the same graft from rabbit 2. We give it a graft from rabbit 2. The graft is accepted. What can we conclude? Since in the absence of mature T cells due to the ablation of the thymus, the non-self graft was accepted. Then, who is responsible for the graft rejection? No thymus, no mature T cells. So T cells, mature T cells, they are responsible for the rejection of the graft. Last question. Referring to your acquired knowledge, give 3 morphological traits of T lymphocytes. They have large round nucleus, they have T cell receptors on their membranes, and they have a small cytoplasm, or it's called diffuse cytoplasm. Study well, guys, and good luck.
Here we move to the specific immune response. As we know, the first line of defense in our body is natural barriers. Second line of defense is the non-specific immune response. Why? The second line of defense. What happens when the second line of defense fails? Third line of defense appear. This is called the specific immune response. So first, we have the first line of defense, which is the natural barriers. Second line of defense represent the non-specific immune response. So what happens when the second line of defense fails? We have a third line of defense, which is the specific immune response. What is the specific immune response? Specific immune response. It is working in the case of the activation of T lymphocytes. We have cases of the activation of T lymphocyte, they are cases of viral infection, and we have steps of the activation of T lymphocyte. First,
the specific T lymphocyte approach the target or the infected cell. Step number 2, the T lymphocyte, they adhere, they bind to the target cell or the infected cell. They bind to it using the T cell receptor, they are called the TCR. And whenever they adhere, the T lymphocyte give the target cell a killer kiss. It perforates, meaning that it creates holes, it creates pores. As such, the target cell dies. This is an important point. Be careful to this. This is an important part to work on or to study. This is an important note. Please note this. Take this into your consideration because you are going to have such a question in your exam. First stage, this is the T lymphocyte working. It starts approaching the target cell. Second stage, it adheres to it. Third stage, it releases perforin to make pores or perforate the cell. Case of activation of T lymphocyte, the second case is the case of graft rejection, mainly the allograft. So this is the T lymphocyte attacking a cancer cell. This is a figure showing a T cell, a T lymphocyte attacking a cancerous cell. So here we move to the type of the immune response carried out by T lymphocyte. We have something called the specific immune response. So what is the type of the immune response carried out by T lymphocyte? Is specific. Name of the immune response carried out by the T lymphocyte, it's called the cell-mediated immune response. So all the immune response is called specific cell-mediated immune response. So therefore, T lymphocytes carry out specific cell-mediated immune response where the T cells or T lymphocytes kill cells in both cases. Here you have to stop. What about the blood groups? Blood groups. Remember, red blood cells, they don't have a nucleus. They are enucleated cells. As such, they don't have HLA on their surfaces. So here we have antigen A, antigen B, antigen A and antigen B, and we have here no antigens. So this is a blood group A. And this is blood group B, this is blood group AB, and here we have no antigen, so it's blood group O. This is a revision for blood groups. Blood group A has antigen A, blood group B has antigen B, blood group AB has antigen A and antigen B, while blood group O, it has no antigens. So here, the antibodies present in the serum. Blood group A, it has antibody B in the serum. Blood group B, it has antibody A in the serum. Blood group AB, no antibodies. And blood group O, it has antibody A and antibody B. Note, antigen AG, symbol for the antigen, or it's called agglutinogen. If you see both words, antigen or agglutinogen, the same, while AB, it calls for antibody or agglutinin. Application. If we add antibody A to blood sample of group A, what would happen? If we add antibody B to blood sample of group A, what would happen? If we add antibody A to blood sample of group AB, what would happen? And also, if we add antibody A to blood sample of group O, what would happen? First one, if we add blood group A, we add antibody. The red blood cells of a person of blood group A carry antigen A on their surface. So upon adding antibody A to this blood sample, antibody A can move toward the antigen A, so this called agglutination. Agglutination is the binding of an antibody to its corresponding antigen. While if we add antibody B to blood group A, so blood group cells of blood group A carry the antigen A on their surface. Upon adding antibody B can't bind. No binding between antibody B and antigen A. So they can bind, so no agglutination occurs. While the red blood cells of a group of blood group AB carry both antigens, antigen A and antigen B on their surface. So upon adding antibody A can move toward antigen A. Antibody A binds to antigen A, leading to agglutination. The blood cells of a person of blood group O do not carry any antigen on their surface. So upon adding antibody A to this blood sample, antibody A cannot bind any antigen, thus no agglutination occurs. Blood transfer. Be careful to study the possibility of transferring blood from a donor, the person who donates, to recipient, the person that received the blood sample. We must study 3 important notes. 1. The antigens found on the red blood cells of the donor, the antibodies found in the serum of the recipient, and you have to know that no agglutination should occur for the blood transfer to be accepted. This is the same application of donation of blood. The donor, if it's blood group A, so his or her blood cells carry antigen A on their surface. The recipient is of blood group B, so he or she has antibody A in the serum. So agglutination occurs and the blood transfer is not accepted. While if the donor is of blood group A, his or her red blood cells, they carry the antigen A on their surface. The recipient is of blood group AB, so she has no antibodies in his serum. Upon blood transfer, no antibodies in the recipient may bind to the antigen, so no agglutination. And most important, the blood transfer is accepted. Blood group determination. This showing 4 samples. We have 4 samples in the lab. We don't know for whom is these samples. We have to interpret. Be careful. It's analyze plus an indication. So you have to know this, analysis plus indication. So after adding antibody A and antibody B to blood sample 1, agglutination occurs in both cases. So this indicates the presence of both antigens, antigen A and antigen B, so the blood group is AB. While after adding antibody A and antibody B to blood sample 2, agglutination occurs only with antibody B. This indicates the presence of antigen B and not antigen A, so the blood group here is B. While after adding antibody A and antibody B to blood sample 4, no agglutination occurs. So no antigen A, no antigen B. This indicates that this blood group is O. The donor is of blood group AB, so his or her blood cells, they carry antigen A and antigen B. The recipient is of blood group B, so he or she has antibody A, so there is an agglutination since antibody A can move toward antigen A and they will agglutinate. So this blood transfer is not accepted. Okay, this is the same one as we talked before. Here are the cases of the activation of B lymphocyte. First, case of blood transfusion, this activation of B lymphocyte. 2, the case of bacterial infection. This is a green antibody battles a red antigen. This is figure showing an antibody, which is the green antibody battles the red antigen. Antigen-antibody complex, this is showing antigen and antibody binding. This is a unique chemical fit. Once infected, the antibody memory is stored, and immunity or protection when invaded again. This is called the memory cells. Type of immune response carried out by B lymphocytes, it's called a specific immune response. What do we call the name of the immune response carried out by B lymphocyte? It is called the humoral immune response. So B lymphocyte, they carry out a specific humoral immune response where B lymphocyte, they secrete what's known by antibodies. They are called antibody secreting cells against the non-self into serum in both cases. Keep in mind, primary response, secondary response. What is the primary response? It's the immune response upon the first contact with the pathogen. the first contact with the pathogen, it's called the primary immune response. While the secondary immune response is the immune response upon the second contact with the same pathogen. Be careful, the same pathogen, not a different pathogen. So the primary immune response is the immune response upon the first contact with the pathogen, while the secondary immune response is the immune response upon the second contact with the same pathogen. This figure shows, first we have antigen recognition, number two we have something called multiplication, and we have antibody secreting cells, and we develop something called memory cells, and three, they differentiate either antibody or memory cells. These are effector cells. This is the amplification of B lymphocyte. The figure just shows the amplification of B lymphocyte with developed plasma cells or memory cells. Keep in mind, proliferation, it means amplification. Antibody secreting cells, they are called plasma cells. During differentiation, a cell differentiates into effector cells and memory cells. Effector B cells, they secrete antibody, they are called plasma, and effector T cells, they are called the killer cells. Pay attention to this graph. This graph shows the injection of an antigen or the variation of the amount of antibodies in arbitrary unit AU as the function of time in days. Upon first injection of the antigen at time equal 0, the time taken is called latency. It's about 8 to 10 days to start secretion antibody, where the maximum amount of antibodies is here about 2 AU, 2 arbitrary units. The time is this one, so about 30 days. While upon the second injection, here, the second injection with the same antigen, the secondary immune response is more fast, it's faster, so it takes about 2 to 3 days to start an immune response and secrete a maximum of 6 AU, 6 arbitrary unit of antibodies. If you see here, this is the primary, and this one represents the secondary immune response. This one, long latency period, represent 10 days, while here is short latency period, about 2 days or 3 days maybe. We have low amount of antibodies, 2 AU, while here we have a high amount of antibodies, about 6 AU. And also this short duration of protection, about 10 days, 30 days, sorry, approximately, short the duration of response or protection, while here is a long duration of protection or response, about 40 days approximately. So this figure, the same figure or another form, first injection, primary response, low amount of antibody, short duration and long latency, while secondary immune response, long duration of protection, short latency period, and a high amount of antibodies. The difference between the primary and the secondary. Primary immune response regarding the latency, which is the time needed to start the immune response, amount of antibody, and duration. Whenever you have to draw any table in the exam, you have to give it a title and you have to draw it using a pencil and close it properly. So primary immune response, long, about 12 days, while the secondary is short, 5 days only. Amount of antibody is low, amount of antibody in the secondary, they are high. Duration, it's short, duration here it's a long one. Keep in mind that the primary immune response takes a long time to eliminate or to destroy the infection, yet it produces specific memory B and T cells needed for subsequent invasion, while the secondary immune response takes a shorter time and is more efficient due to the presence of memory cells. Be careful to this point. Be careful to this point. Secondary immune response takes a shorter time and is more efficient. Why? Due to the presence of memory cells. From where? From the primary immune response. This is very important. Also, this is the figure, first exposure to the antigen and the second exposure. This is called the primary immune response, and this is called the secondary immune response. Secondary immune response is more efficient due to the presence of what? Of memory cells. From where? From the primary immune response. To be careful to this point, study well for your exam. The exam is so easy. Just study well. Take care of the notes that I said here and the notes in the class, and you'll do well. I'm sure of this. So good luck, guys. Bye bye.